Hello students, this is the lesson video for lesson 4.02, the second lesson in Unit 4, and we're going to be talking about Mendelian inheritance. Here's a general overview of what we'll be going over in this lesson. We're going to be focusing on the concept of Punnett squares and how they are used to predict offspring genotypes and phenotypes. Both of these things we will define. And they're also used to infer parental genotypes as well. So we can predict future offspring, and we can also try to figure out what genes a certain parent has as well. Okay, so the goals for the lesson is the same as the last lesson, where we want to know the genetic basis for Mendel's laws of segregation and independent assortment. We also want to understand what a gene is and what an allele is. You should be able to explain the fundamental rules of inheritance through the work of Gregor Mendel, and also how they have slightly changed since their initial discovery. You should be able to describe Mendel's experiments that led to the laws of segregation and independent assortment. And the last thing is you should be able to distinguish between dominant and recessive traits. Okay, so before we go into how exactly we use Punnett squares in genetics, let's go ahead and review meiosis. So in meiosis, the really key takeaway is that alleles segregate during meiosis. Remember, alleles are different versions of the same gene. Okay, so say that this was one of your cells. You basically get one, let's say the pink ones are from your mom, and the blue ones are from your dad. Okay, and let's just say that this is chromosome 1 and chromosome 2. Okay, so you have two copies of chromosome 1, one from your mom, one from your dad, and the same thing for chromosome number 2. You have one from your mom and one from your dad. So this is in a normal body cell. But if you look at the end, so the end result of meiosis, what we end up with is you only have one version of each of the chromosome in each of the resulting cells. Okay, so here you had two of them, and here you only have one. Okay, so when gametes, which are basically sex cells, so this could be sperm or egg cells, formed during meiosis, pairs of homologous chromosomes carrying alleles separate. Okay, so pairs of homologous chromosomes. Homologous chromosomes are basically the same genes. Okay, so homologous chromosomes have the same genes, but one is from your dad and one is from your mom. Okay, so these two right here that I'm circling, these are both chromosome number 1, but one is from your mom and one is from your dad, and we call those homologous chromosomes. So during meiosis, the pairs of homologous chromosomes will separate. Okay, so that at the end, each gamete or sex cell receives only one allele per characteristic or per gene. The homologous chromosomes pair up during prophase 1, and one chromosome from each pair ends up in a new cell after cytokinesis. Okay, so the first thing we know about meiosis is that in the first round, we have separation of homologous chromosomes. Your mom and dad's chromosomes are separating. Okay, so this might seem like a lot of information, so let's go ahead and quickly review this information by going over meiosis 1 and 2. So here we have each step of meiosis 1 and meiosis 2. How you can tell the difference between the two phases is that we have, as you can see, a 1 after each of the phases, and then the phases after that you see right here will have 2 afterwards, and so that means you know that it's part of meiosis 2. Okay, so the first phase is we have interphase, and this is before meiosis actually begins. What's happening here is the DNA is replicating inside the nucleus, and so what we now have are 2 copies of every single chromosome. And then now what you see is we have in the nucleus we have chromosomes, right? So the DNA is no longer stringy looking, it becomes visible, and the reason why is because it's actually condensing. Another thing that you see is the nuclear envelope, which is basically the surrounding of the nucleus is going to start breaking down so that the chromosomes can now exit the nucleus. One really cool thing that happens during prophase 1 is that there's some crossing over as well. Okay, and what crossing over means is it's basically, as you can see right here, we have homologous chromosomes, like one from the mom and one from the dad, same genes but different alleles, are crossing over. So they're exchanging information, genetic information, so that it increases variation in your sex cells. Okay, so you see that happening right here and also right here. So that all happens during prophase 1. Okay, the next phase is metaphase 1. And so what you see here is we have spindle fibers that form in the cell. And the spindle fibers actually arrange these chromosomes so that they're aligned in the middle of the cell. And that's why we call it metaphase. It's in the middle of the cell. And so what you see here is that pairs of homologous chromosomes. Remember, those are the chromosomes that have the same genes, one from your dad, one from your mom. And you see that they have paired up next to each other. Okay, and then the next phase is called anaphase. And clearly what's happening right here is that in anaphase, the spindle fibers are going to pull one of the homologous chromosomes from each pair to the opposite poles of the cell. So now we have the separation of chromosomes. So for this chromosome, dad's chromosome went to this side, whereas mom's went to the other, and vice versa for the other chromosome. So now we no longer have homologous pairs, they have separated to opposite ends of the cell, and that's anaphase. Next, what you see is we have telophase 1 and also cytokinesis. This telophase is when you then have the nuclear envelope forming again, so the chromosomes are now within that nuclear envelope, and the cytoplasm divides during cytokinesis, okay, and that's like all the other surrounding area of the cell. And so clearly after this cytokinesis happens, where you see this cleavage furrow, we will now have 2 distinct cells instead of just 1. The next phase is called prophase, okay? So prophase 2. So you see here we have a 2 afterwards, which means that it's part of meiosis 2. And it's the same exact thing happening. We have the nuclear envelope we see is going to start breaking down, which will release the chromosomes into the cells. And we have those spindle fibers that we used in order to align and separate chromosomes within the cell. The nuclear envelope disappeared. We have the spindle fibers putting the chromosomes in place, right? So now we have metaphase 2, where we have the chromosomes lined up at the equator and the spindle fibers attached to the centromeres of each chromosome. Okay, so instead of having 2 copies, remember we only have 1. And so instead of homologous chromosomes separating, since that already happened, we now have chromatids separating. So basically, one side will go to the left part of the cell and the other side will go to the right side. So it'll be pulling in the same way. So anaphase 2, this is when the spindle fibers pull the chromatids apart at the centromeres and the chromatids move to those opposite ends. And then after it moves to opposite ends, we now have the last 2 phases. Telophase 2, which is when we have that nuclear envelope that encloses the DNA once more. We also have cytokinesis. Remember, that's really important because that's when we divide up that cytoplasm. And our result is we have 4 cells and each of them have 1 copy of each gene versus having 2 like the beginning of meiosis. Okay, so alleles, remember, are versions of a gene, right? So if the if we were talking about the eye-colored gene, an allele, for example, would be blue eyes, green eyes, brown eyes, etc. Remember that alleles on chromosomes in an organism determine the physical traits. So depending on which allele you will have different physical appearances. So let's look at Mendel's purple-flowered pea plants that is heterozygous. Remember heterozygous, hetero meaning different, right? This flower has 2 different alleles. And so if we were to look and zoom in into the plant's chromosomes, what we would see is that this plant has a chromosome, one for purple, right? And the other one would be for white. So when this plant makes its sex cells, it will then distribute one allele, so one purple gene and one white gene, into each of the cells so that they only possess one copy for each gene. So they'll either have the allele for purple flowers or the allele for the white flowers. Alleles for different characteristics assort independently during meiosis. So remember, this is a very important concept that Mendel discovered of independent assortment. And so if you could observe meiosis inside the pea plant cells, you would also see that when the alleles for one trait, such as flower color, separate into different gametes, so let's say like a purple allele in one, a white allele in another, alleles for other traits such as pea pod shape or seed color will separate as well. However, they do separate independently. For example, just because a pea plant has purple flowers, that doesn't determine what the pea pod shape would be or the seed color would be. All of these different genes separate independently of one another, and that's the concept of independent assortment. So when chromosomes move into gametes or into those sex cells during meiosis, the alleles they are carrying move with them. Thus, alleles for different traits carried on different chromosomes can combine in many ways in the gametes. And so what that means is you can have a pea plant with purple flowers with green seeds that are wrinkled, or you can have one that has white flowers with wrinkled seeds and yellow seeds. And so the possibilities are really endless because each of these characteristics are going to divide independently during meiosis. Okay, so now we're going to watch a video on Mendel's pea plants, and so we're going to be looking at how he used a method of fertilizing many generations of pea plants to discover their patterns of inheritance. And so we're going to go ahead and take a look at that in this video.
Mendel used a painstaking method for fertilizing his plants, transferring pollen from the male part of one plant to the female part of another.
Oh, you're castrating it.
Absolutely.
So then we go to the other flower,
Yeah, which is over here.
which is the red one.
Pull that off.
Put it off.
Oops.
God, it's not very kind, is it, this
Oh.
sex business?
No, it's rough and ready. Here we go. Right.
And this is how Mendel
Pull
did
the-
it, is it?
Yes, same techniques really. Right. So this time we use the style of this one, which has already got pollen on,
Yeah.
and use it as a paintbrush. You apply it. That's it. Sex in peas. And there we
And
are.
that's it. That's it.
Mendel's research was incredibly thorough. Over 10 years, he studied more than 20,000 plants and kept detailed records. He bred generation after generation of peas, and he looked at how the various characteristics cropped up. Eventually, he noticed a consistent and remarkable pattern.
What Mendel discovered was really weird. He took an absolutely true breeding red-flowered pea. He'd bred it for generations and it was always red. And he took also a true breeding white-flowered pea. And then he deliberately crossed these. And what he got was only red peas. It looked as if the white had been lost. But he didn't give up there. He let these ones self-fertilise. And in the next generation, well, it began to look as if the white had been lost. But then up popped the white again. And he bred thousands of them, and he got consistently a 3-to-1 ratio of red to white.
Mendel had discovered a fundamental law of inheritance. When plants breed, each parent passes on a factor, a set of instructions for creating every one of the offspring's physical characteristics. Only one of these factors is activated, but the other factor lies dormant and can reappear in later generations.
So now let's go over genotype versus phenotype. So the combination of alleles or versions of a gene in an organism is a genotype. Scientists refer to the alleles associated with different traits using uppercase and lowercase letters. So in general, the rule is that an uppercase letter represents an allele that is dominant over another trait. The trait that is not dominant is called recessive, and its allele is represented by a lowercase letter. So the allele for purple flowers and pea plants, for example, is dominant. Okay, so purple is dominant, so that we have P, big P, which would be for purple. And let's say that it's dominant. Okay, and then for the white flowers, we'll have a little p for the white gene, and this one is recessive. So even when a plant has the allele for purple flowers, so a big P, and an allele for the white flowers, which is a little p, what you actually see is just a purple flower. And the reason why is because that big P, or that purple allele, dominates over the white allele. So all you see is the purple color. Remember, in the previous lesson, many scientists believed that if an organism had both alleles, that it would be maybe a lighter purple, using the blending inheritance hypothesis, but Mendel discovered that this was not correct. So these two letters is what we call a genotype, and it basically tells you what allele an organism has for a certain gene. So for example, for this gene of flower color, we can have 3 different genotypes. A plant could have 2 big P's, a big P, a little p, and also a little p, little p. Obviously, we can't look at a flower and know exactly which genotype it has in every single case, but if you do know the genotype, it will tell you what the phenotype is. And phenotype is basically what physical appearance results from having a certain genotype. Okay, so knowing the organism's genotype will help you predict its phenotype or physical appearance. So again, just to define phenotype is an organism's observable characteristics, so what you can physically see. So when you see a pea plant and you see these purple flowers, you are seeing the phenotype. You're not seeing the genotype, but you're seeing the phenotype. An organism's phenotype is determined partly by its genotype and partly by its environment. So can you use a plant's phenotype, or remember this is physical appearance, to determine what genes it has in its genotype? Not always. So for example, if you were looking at a purple flower, you wouldn't know whether its genotype would be big P, little p, or big P, because both of them would result in having the same phenotype, which is that they would have purple flowers. If a pea plant has alleles big P for flower color, and also has 2 different alleles for the pea pod shape, So for example, big R would be for rounded pods like you see right here, and then little r would be the constricted pods. You can write that plant's genotype together. You could do the big P, and big R, little r. Knowing the plant's genotype, you would predict that its phenotype would be purple flowers because of the big P, big P. Remember, big P is purple. And that it would have rounded pea pods. Why? Because it has that big R. So you would know that it had purple flowers and rounded pea pods. However, if we had a genotype where they're all lowercase letters, which means you know that all of the traits for this plant would be recessive, we know that it would result in white flowers and constricted pea pods. And so again, by knowing these genotypes, you can then predict what the phenotype or physical appearance would be. Okay, so now that we know what a genotype and a phenotype is, and we also know the difference between dominant and recessive traits, we can now go into the main part of this lesson, which is the Punnett square. So what a Punnett square does is it helps you predict possible genotypes of offspring based on the parents' genotypes. Okay, so again, we take the parents' genotypes and it helps us predict what the offspring would look like. So plant and animal breeders need to be able to predict the characteristics of the offspring of a given set of parents because they want to create a certain result. So one tool they can use is this Punnett square. And it really is useful because it shows all the possible ways in which alleles from both parents may combine. So it can help predict the probability or the frequency of offspring genotypes based on parent genotypes. A Punnett square uses the fact that alleles separate during gamete formation and that new combinations of alleles are created when an egg and sperm join. And so we use the concept that Mendel discovered of independent assortment, knowing that all of these alleles that we're talking about are going to separate independently during meiosis. So this Punnett square right here shows the possible offspring of two purple flowered pea plants that are heterozygous. Remember, hetero meaning different. So they have one big pea and one little pea. One big pea and one little pea. So this is a Punnett square that predicts what its offspring would look like. And just as Mendel observed, we have a 3-to-1 ratio. We have 3 of the purple and 1 of the white. Okay, so how exactly do we actually use these Punnett squares? So again, the Punnett square basically is showing you all the possible different allele combinations and genotypes of an offspring based on the genotypes of the parents. So I'm sure all of you guys have encountered fruit flies before. They actually have many different eye colors. 2 of the most common ones are red eyes and brown eyes. So red eyes is dominant, so we're going to say it's big R, and brown eyes is recessive, so we're going to say that it's little r. So let's go ahead and take that example and cross 2 of these fruit flies. So let's say that we're crossing a homozygous, homo meaning the same, so has 2 of the same alleles, red-eyed fly. So that means if it's homozygous, has the same 2 alleles, and it has red eyes, it must be big R, big R. And so that you see that right here. And so what we're going to do is we're going to take each of those R's and put it into separate sections, because remember we have that independent assortment. It's going to separate 2 of its alleles into separate sex cells. And then we have a homozygous, which again means same, brown-eyed fly, so that would be little r, and we're going to separate that into 2 separate sections as well because of the concept of independent assortment. After that, what you're going to do is you're going to just draw a Punnett square, which is really simple. Just looks something like this, right? So that you have 4 squares to fill out in the end. Okay, again, remember homozygous is kind of the same thing as saying true breeding or purebred. It only has one particular version for each gene, one particular allele for a certain gene, and so it's always going to produce offspring that have the phenotype for that trait when it is crossed with itself. So next, since we have the sides, remember we separated them. Once we have the sides completed, we can now fill out the Punnett square. And so how you fill it out is you basically take whatever you have right here, the top, and you're going to go ahead and distribute it across. Okay, so we'll take that big R and we'll bring it one to this square, one to this square, this other big R, one to this square, and the other one to this square. And we'll do the same with the little r's. The only difference is that instead of going across, we're going to go down. So we have to make sure we do it to the top one and also the bottom, and the same thing here. Okay, and so the resulting genotypes that end up is that you have 2 alleles for each offspring, right, which is correct. You should have 2 in each box. And all of them have the same genotype. All of them have one big R and one little r. So we have 100% of the same genotype. And remember, this is the same conclusion that Mendel came up with when he was crossing his pea plants. Okay, so all of them resulted with red eyes. Okay, so just like we talked about before, when you have a certain genotype, you can then predict what the phenotype or the physical appearance of an organism would be. So again, genes from a genotype, right, or actually it's alleles of genes, allow us to predict the physical appearance. Okay, so if we look at the Punnett square we filled out right here, remember we had a red-eyed fly with a brown-eyed fly, and we crossed them and we saw that we had all big R, little r, big R, little r, big R, little r, and big R, little r. And so because we know the possible genotypes, we can then predict the phenotype. And we know that the big R, right, is dominant. So if the big R is dominant, it's going to dominate over the recessive allele. And so what that means is that all of the big R, little r will have red eyes. Okay, so this would be the phenotype, and this would be the genotype. So you can see really simply here how the genotype helps us predict what the phenotype would be. A Punnett square also can help you predict the traits of future generations. So even if you haven't crossed certain organisms yet, you can still predict the traits of future generations. And so remember, we created that first generation, which was that F1 generation, when we crossed the big R, little r, and they're all heterozygous, right? If we were to take 2 of those flies from that generation, so big R, little r, big R, little r, and cross that, we would have a different result. And so remember, we separate each of the alleles into different sections, right? So we have a big R here, a little r here, and the same thing right here. And then we make sure that we distribute all of our alleles properly, right? So this big R right here, we would put it all the way down. Little r, we'd bring it all the way down here. And the same thing, big R all the way across, and then the little r all the way across as well. And what we end up with is we have one big R, we have 2 big R, little r's, and the last one we have a little r, little r. Okay, so what that really shows you is that we can use the Punnett square to really come up with different percentages or the probability that certain offspring of 2 parents will have a particular trait. Okay, so probabilities are usually described as percentages, ratios, or fractions, and you'll have to do some form of that in your assignments. In genetics, probability determines the likelihood that a specific allele will occur in a gamete. It also determines the outcome of genetic crosses. So for example, what is the probability that the offspring of a homozygous red fly and a homozygous brown fly will have red eyes? In that case, remember, we found that it was 100%. So the probability of the outcome is 1, because all of them will have red eyes. However, if we looked at the cross we just did for the F2 generation, we see that one fly has the genotype big R, the other two have big R, and the last one has little r. And so these right here, they all have red eyes because they have that big R allele. However, this one right here is going to have brown eyes. So instead of having that 100% ratio that we saw before, we now see that the probability of offspring with red eyes is 3 out of 4. That's a fraction. Or we can write as a ratio, which is 3 to 1. Okay, so the overall thing that you should have understood from this lesson is how to use a Punnett square to predict offspring's genotypes, which remember are the alleles, right, of certain genes that an individual has, and how that can help us predict phenotypes, which is the physical appearance as well. We can also use Punnett squares to infer parental genotypes as well, which means we can actually predict what the parents' genotypes are if it's not clear. For example, remember we looked at those purple flowers. Sometimes the purple flowers can have big P, or big P, little p. A Punnett square can help us determine which one it most likely would be. So here's some important vocabulary. We have alleles, a concept we've gone over already, which is a specific form or version of a gene. We have heterozygous, which means that you have 2 different alleles for a gene. We have dominant, which means that an allele is expressed even if it only has 1 copy. Remember, it's just like having that big P, little p. The big P is dominant, so we're going to see whatever the big P trait is. We have recessive, which is the characteristic or the allele associated with the trait that is only expressed if you have 2 copies of that same allele. So that's like if we have a little p, then we'll be able to see that trait. We also went over what a genotype was, which is basically the alleles present for a certain gene. Each organism will always have 2 alleles or 2 letters in their genotype. That's really important for you to know because they always get one from their mom and one from their dad. We learned about phenotypes, which is the physical or detectable traits of an organism. We talked about how the phenotypes is often dictated by the genotype of an individual. We went over how to complete a Punnett square, which is a grid used to predict the results of genetic crosses using the parent's genotype. And then one of the last words is homozygous, which is having 2 identical alleles for a gene being considered.